Welcome to Cell Biology for Pharmacy. This video is the first in a series on the cell cycle. In this video, we will review several components of the mammalian cell that are involved in the cell cycle, namely chromosomes, centrosomes, and microtubules. Let us begin. Before we dive into the cell cycle, we first need to understand how DNA is stored inside the cell. Here you can see several representations of the structure of DNA and how it is stored inside the nucleus, from the double helix structure you can see on the right to the compact structure of the mitotic chromosome on the left. Large amounts of DNA are required to encode all the information needed to make complex organisms, especially eukaryotes. Now, in eukaryotic cells, all of this information is stored in the cell nucleus, which is only about 5 to 8 micrometers in diameter. This is relatively very small when we consider that each cell contains around 2 meters of DNA. This is roughly equivalent to packing 40 kilometers of very fine thread into the size of a tennis ball. Inside the nucleus, DNA are packed in a very condensed manner into structures called chromosomes. But the DNA strands cannot just be stored in any random way. They are packed into chromosomes in a very structured fashion, as you can see here in this diagram. Specialized proteins, shown here in yellow, are involved in the folding and packing of the DNA. These proteins bind to and stabilize the structure of DNA, and it generates coils and loops that prevent the DNA from becoming entangled in a mess. In eukaryotes, not all of the DNA inside a cell is in the form of one extremely long strand. DNA in the nucleus is distributed to a set of different chromosomes. For example, the DNA in a human nucleus contains around 3.2 billion nucleotides, which are divided into 23 different types of chromosomes. All human cells, except for the sex cells, which are the sperm and egg cells, and red blood cells, which do not have a nucleus, contain 2 copies of each type of chromosome, resulting in a total of 46 chromosomes. Now, each copy of the different chromosomes are inherited from the father and the mother. In this image, you can see the 23 different types of human chromosomes, which have been colored using fluorescent dye. Now, you may think that because the chromosomes carry such a large amount of information, all of that information is essential. However, that is not the case. Large segments of DNA are actually quite useless or have unknown functions, and only small portions of it carry useful information and/or perform certain functions. Segments of DNA that contain instructions for making particular proteins or RNA molecules are called genes. In this diagram, you can see that only certain segments of yeast DNA are genes, which are colored pink, and the rest of the DNA, represented in gray, either do not have any function or their functions are still unknown. Other than carrying the genes, it is also very important that chromosomes are able to be duplicated. This is crucial for cell division, because each resulting daughter cell must have the exact same copy of the DNA. Now, during interphase, gene expression, which is the process of creating proteins and RNA from DNA, occur continuously. Hence, the DNA are in the form of structures called interphase chromosomes. Interphase chromosomes are unfolded, very long, very thin, and tangled threads of DNA. This structure allows the different enzymes that are involved in DNA replication and gene expression to recognize and bind to the DNA. However, during cell division, the interphase chromosomes condenses into mitotic chromosomes, which have a very dense structure, as we've seen previously. If we observe a cell under the microscope, most of the time we will not be able to see any activity. This is because the cell is in interphase, a period of time in which the cell is not dividing. During this time, it is impossible to distinguish between chromosomes, as you can see in the picture on the top right. The chromosomes are in the form of interphase chromosomes, which are long, thin, and entangled. DNA duplication occurs when the cell is not dividing. However, during cell division, the duplicated chromosomes are separated to opposite ends of the cell, and the cell splits into two daughter cells, each with a complete and exact copy of the parent DNA. You can see on the diagram here the chromosomes being pulled apart to opposite ends of the cell. As we've seen previously, most parts of DNA are not genes and are actually quite useless, since they have no known function. However, some parts of DNA that are not genes do have specialized functions. There are three main specialized segments of DNA. First, there are replication origins, which are segments of DNA where enzymes that are involved in DNA replication attaches to and begins the replication process. As you can see, eukaryotic chromosomes have multiple replication origins, which allows for faster DNA duplication. Secondly, there are telomeres, which are located at the ends of the chromosomes. These DNA segments allow the correct replication of the whole chromosome and prevents the DNA from being mistaken for damaged DNA. And thirdly, there is the centromere. The centromere allows the duplicated chromosomes to attach to the mitotic spindle and be separated to opposite ends of the cell during cell division. Now, during cell division, the DNA is coiled up and packed very densely, forming a compact structure called the mitotic chromosome, as you can see here. These mitotic chromosomes are easily visualized, and different types of chromosomes can easily be distinguished. At the center of the mitotic chromosomes are the centromeres. The centromeres allow the chromosomes to attach to a structure called the mitotic spindle, which helps split apart the duplicated chromosomes to opposite ends of the cell. The mitotic spindle is a structure that is essential in cell division. Without it, cell division would fail and the cell would die. Now, this structure is made up of microtubules, which are long, hollow tubes made up of proteins called tubulin. Microtubules are polymers that are made up of tubulin subunits. There are two types of tubulin proteins that make up microtubules, namely alpha and beta tubulin. You can see on the diagram shown here how the tubulin subunits form the microtubule structure. Other than the mitotic spindle, microtubules also make up the cytoskeleton, which is also an important part of the cell. The cytoskeleton provides the cell physical support and gives it strength and its shape. Another important structure involved in cell division is the centrosome. Tubulin, the proteins that form microtubules, polymerizes or grow from nucleation sites on centrosomes. The centrosome is the major microtubule organizing center in animal cells. They control the location, the number, and also the orientation of microtubules that grow from their nucleation sites. The centrosomes are typically located near the nucleus when the cell is not dividing and move toward opposite ends of the cell during cell division. That's the end of this video. Next, we will discuss the eukaryotic cell cycle. Thank you.